So, a double devil dilemma. No, dilemma. A double devil dilemma. Say that quickly and you'll get as tongue-tied as I just was. But that's the title of our address today from Emeritus Professor Greg Woods, and he is eminently qualified to do so. He's had a lifelong interest in immunology and studies led to studies of T cells, leukaemia, UV irradiation, tumour immunology, dendritic cells, and particularly Devil Facial Tumour Disease. His research career includes laboratories in Toronto, London and Scotland. As an academic, he's had leadership roles and taught immunology to undergraduates and postgraduates. His awards include a Eureka Award for Environmental Science and the AMSA Research Award in recognition of distinguished service to medicine, science and the community. Following his retirement in 2017, he was bestowed with the status Emeritus. So it's my very great pleasure to welcome to the dais Emeritus Professor Greg Woods to talk to us about a double devil dilemma. Greg.
Thank you for the invitation, and thank you everyone for coming today, either directly, which is lovely to see faces, lovely to see whole bodies actually, those on Zoom and those who'll YouTube it later. If we can get the first slide going, will be a double devil dilemma. It is hard to say, isn't it? I didn't realise how hard it was to say until I actually say it out loud. I've called it that title because of a number of things that will come up, and they'll come up obviously. Okay, I first want to talk about Taraba. Now I'd like to acknowledge the original inhabitants of this land, either the elders past, present and future, and just briefly talk about Taraba. Taraba was a beautiful black animal, and being so beautiful, he was very lazy. And for his meals, what he used to do was when the parents would go out, he would steal the young and eat the young. Now, this was not very well appreciated, as you can appreciate. One night, he got caught by the spirit helpers, and they punished him. His beautiful curly fur coat was made straight, his beautiful round snout was made pointy, he had little ears and little eyes, and his voice was changed into a horrific growl so that you would know he was in the neighbourhood. And as he was leaving, he was sprayed with white clematis leaves, and which gives him the white stripe on the chest and on the rump, so it could be easily seen. When the Europeans arrived, they heard this growl in the wild, and of course they assumed it was the devil, hence the name Tasmanian devil. The scientific name is Sarcophilus harrisii. Another nice one to get your tongue around. Sarcophilus means meat lover, obviously for they love meat, and harrisii comes from the Surveyor General who used to sketch Tasmanian devils early on in Tasmania's history. So what I'm going to talk about today is what is DFTD, Devil Facial Tumour Disease. What is DFT2? You may not be aware of it, but there is a second cancer. Why aren't the cells rejected as allografts? I'll give you a bit of background on our vaccine trials, why devils are susceptible to DFTD, talk about some new technologies, and I'll apologise in advance, there's going to be some fairly scientific slides. I'll try to explain them carefully, but I'll give a clear conclusion at the end, and some future research. Since I retired, Bruce Lyons took over the lab, and he and I worked together to try to understand the immune response of the devil's immune system with devil facial tumour cells. Why aren't they rejected? And hopefully to develop a vaccine. Currently in the lab is Dr. Andy Flees. He's running the lab. He's an excellent molecular biologist and is introducing new technologies. Most of the work early work I'll be talking about is with Alexander Kreis and Cesar Tovar, who did the early work at understanding the immune system. Ruth Pye did a PhD with us, is a vet, and she was the one who discovered DFT2. And two young PhD students, Camilla Esposito and Amanda Patchett, introduced some of the bioinformatics. So I'm going to go from the old to the new technologies. The devil dilemma has many risks: habitat loss, roadkills, feral cats, and DFTD. I'll talk about roadkill a little bit later. So the devil and his cancer, estimated 1996, about 30,000 devils roam the state. And as you all know, the Tasmanian devils only exists in Tasmania. When the Bass Strait flooded, we were cut off from the mainland. Devils on the mainland became extinct, and they only survived within Tasmania. Gradually, in one devil, I still can't believe this, one devil had one cell that mutated and became a cancer cell. That grew into a cancer and was transmitted from devil to devil to devil throughout the whole of the state, killing every single devil it infected. If you got infected, there was only one outcome, was death. It hasn't quite reached the far northwest or the far southwest. Obviously, the reason the southwest, it's not very good devil habitat. And then the population diminished. In 2014, incredibly, we discovered a second transmissible cancer. Transmissible cancers are extremely rare. We don't catch cancer. They are rare. In the mammalian population, there are two transmissible cancers. One is in dogs and one is in devils. Now devils have got two. They must be extremely unlucky to be struck by two transmissible cancers. And latest results indicate that we've got about 74 devils have been trapped, 150 tumours with this second cancer, which we call DFT2. It's restricted to the channel at the moment and hasn't spread any further than that, probably because of the rivers and the mountains. What is DFTD? Well, DFTD is Devil Facial Tumour Disease, and I'm going to call the first cancer, the one discovered in Mount William, DFT1. I'm going to call the second cancer DFT2. That some people call the first cancer DFTD, but I'm going to distinguish. DFTD, I'm going to describe the two cancers, DFT1 and DFT2, and the first one DFT1, the second one DFT2. It is a cancer. It can be found inside the mouth, on the outside. It is a horrific cancer. If anyone's seen devils with devil facial tumour disease, it is horrible. The devils will die from metastases or won't be able to eat. It is a true cancer in every sense of the word. DFT1 is a cancer of Schwann cells. Schwann cells are cells that coat peripheral nerves. In humans, Schwann cell cancers are really, really rare because Schwann cells don't divide, so they don't develop into cancers. But remarkably, the Tasmanian devil has a cancer of Schwann cells. And I'll just highlight, this is a picture of a cancer, normal pathology section, H&E, and along here is a marker called periaxin. Periaxin is a molecule found on peripheral nerves, found on Schwann cells. It is an excellent marker for us to identify the cancer, which is here. So periaxin is an excellent marker for us to identify DFT1. Why have I mentioned periaxin? That will become obvious in a couple of slides. So we've got two, that will become obvious on this slide. We've got two cancers, DFT1 on the left and DFT2 on the right. Underneath, you'll see the periaxin staining that first cancer, nice and brown. Brown tells you that's positive. That tells us that those markers are expressed, and that's DFT1. We found a second devil in the channel region, and that's the devil there that had a tumour. We assumed it was DFTD, which we now call DFT1. But when we stained it with periaxin, it was negative. So we thought, oh, what is this? So when we had to try to work out, is it a different cancer? What is this particular cancer? So we've got a double dilemma. Is it DFT2 or is it DFT1 or is it something completely different? So a technical slide. Basically what we did was we got cell lines from the DFT1, DFT2 cell lines, tumour biopsies, and cell lines from healthy Tasmanian devils. Ran it through this machine called an RNA sequencer, which basically, a fantastic machine which sequences all of the RNA into little fragments and divides it up into all the bases. So you get those particular sequences. We've heard about genomic sequencing of the COVID virus. This is similar, it's genomic sequencing of the RNA. So you can identify different tissues will express different RNAs. For example, your heart will express heart genes, your lungs, lung genes. So if you compare the tumour cells with other tissues, we might get an indication as to what it might be. And I draw your attention to the very right, over here. What you'll notice is where it's red, they're the genes highly expressed, red hot. Where it's blue, where the genes are not highly expressed. To determine what it is, you have to match the reds with the blues. And on that right hand of the panel, you've got DFT1, DFT2, and in the middle, you've got peripheral nerves. So look, but it's very different to every other tumour. So what we concluded from this with a bit more work was that DFT1 was also a Schwann cell, but it was a different type of Schwann cell. So there's different types of Schwann cells, and this is an example of how a Schwann cell develops. It starts off in the neural crest as a pluripotent stem cell and goes through different stages, and then it can become, the DFT1 is a myelin-producing Schwann cells, the cells producing myelin to coat the nerve. DFT2 is a different type of Schwann cell called a repair Schwann cell. When the nerve gets damaged, the Schwann cells become repair Schwann cells and repair that particular damage. So DFT1 and DFT2 are of the same lineage, but different stages of development. So DFT1 is a myelinating Schwann cell. DFT2 is a mesenchymal or a repair Schwann cell. I'll come back to that word mesenchymal a little bit later on. It's just a word, it's a different type of cell. So DFT1, a normal myelin-producing Schwann cell. DFT2 is a Schwann cell that undergoes repair. Why are these significant? I'll come back to that. The next double dilemma is, how can DFT be transmissible or transplantable? A non-technical slide, which you're glad to see, is that their biting behaviour accounts for their transmission. As you know, devils bite each other. And they bite each other during the mating season, and they bite each other over food. And they'll squabble. Males bite males, males bite females, females bite males, and females bite females. So it's spread throughout the population. And they bite each other on the face. That is where the tumour develops, on the face. So they'll bite each other on the face and transmit the tumour cells. So a healthy devil will bite a diseased devil, and if the diseased devil has got a tumour, the tumour will start off in the oral cavity. A diseased devil can bite a healthy devil and transfer the tumour cells to the outside of the face. So the biting behaviour accounts for the transmission. It's quite logical. But why aren't they immunologically rejected? Because an organ transplant, the immune system is so powerful, it gets rejected. This was a dilemma that we were confronted for a number of years. Why does the transmitted cells not get rejected by the devil's immune system? So what we had to do was to analyse the blood of Tasmanian devils, to analyse the immune response. They don't have nice veins to get blood from. Our vets had to do cardiac punctures, quite a delicate technique, but we had experienced vets, and you could get 10, 20 mils of blood quite comfortably, and the devil was quite happy to let us bleed him again. Well, not quite happy, but as long as we trapped him, he was willing. I should add, he was willing when he was anaesthetised to be bled. So we analysed the blood, took it to the laboratory, separated the cells and took out the lymphocytes and did as many studies as we could. And what did we find is that we got lots of work done and we could not find any incapacities in the Tasmanian devil's immune system. They have a competent immune system. The T cells work, the B cells work, the macrophages work, the neutrophils work, their antibodies work, everything works. So there has to be some reason why the tumour was not being rejected. So we moved away from the immune system and started to look at the tumour itself. So an expressed immune system doesn't explain the susceptibility. So what is happening in the tumour? Maybe the cancer cells are producing something that suppresses the immune system, right? Or maybe the cancer cells are basically invisible. The immune system just can't see them. So the answer to that question is complex. We looked at a number of tumours and looked for cytokines which suppress the immune response. So these are two cytokines. That's called interleukin-10 and that's called transforming growth factor beta. You don't need to know what they are. You just need to know that they are proteins of hormones in the immune system will suppress the immune response. And we stain the tumour tissues. Blue represents the tumour cells and brown represents the cytokines. The tumour cells did produce some cytokines that would suppress the immune response, but the levels were extremely low, not enough to explain why the immune system wasn't rejected. The reason why is because the tumour cells are invisible. The tumour cells don't express a molecule we called MHC. MHC is a glycoprotein which is expressed on every single cell. And when I was teaching medical students, if I said MHC, everyone, their eyes would glaze over because it's very complex. But what I can say is that MHC is a molecule expressed on every cell, and those MHC molecules are what the immune system recognises to induce an immune response. If you don't express MHC, you don't express an immune response. And when we looked at that, the tumour cells don't express the MHC molecules, but we looked at the genes of the MHC, and this part here shows you various genes. This is at RNA level. Where you see the white is where the genes are expressed. That's a housekeeping gene. That's MHC class 1. That's expressed. But beta-2M in these TAP1, TAP2 aren't expressed. These molecules are important for making sure the MHC gets to the cell surface. And we looked at flow cytometry. Those pictures there show that the MHC is not found on the surface of the cell. To summarise that, the Devil Facial Tumour Disease don't express any molecules that the immune system can use to recognise, therefore is basically invisible. But what is interesting is this molecule here called IFNG, interferon gamma. That is a hormone of the immune system, and one of its roles is to upregulate MHC. So if we treated the cancer cells with interferon gamma, the MHC goes up. And if the MHC goes up, therefore the cells should go from invisible to visible. And so we did some experiments. We thought we'd immunise devils with these MHC positive cells to see if they induced an immune response. Now, whenever you have a vaccine, you have the antigen, in this case the tumour cells, MHC positive tumour cells, but you also need something to boost the immune response. And they need something to activate the innate immune response. And there's a group of receptors on innate immune cells, your macrophages, your neutrophils, your dendritic cells, called toll-like receptors. We wanted to see whether the Tasmanian devil immune system have these toll-like receptors. And when we looked, we found that just to go back to toll-like receptors, that they were actually found on the fruit fly. It's quite ironical because remember before I said the head of the laboratory at the moment is Andy Flies. And toll-like receptors are found on flies, anyway. So we looked for the expression of toll-like receptors, and basically down the left there, we found that they expressed a whole range of toll-like receptors. So the innate immune cells. So we can activate the innate immune system through the toll-like receptors. We should be activate the specific immune system, T cells, by the MHC positive tumour cells. And so what we did was we immunised devils with sonicated and irradiated MHC positive cells, poly-IC-CpG, which are molecules that bind to the toll-like receptors, and this other substance called ischematrix, which is an adjuvant to localise the infection. So we did 4 trials, 2015 to 2018, immunised all these devils, and nearly every single devil produced an immune response. And this was fantastic news because prior to this, it was almost impossible for the devils to produce an immune response to the tumour cells. So this was promising. So the good news was that nearly all the immunised devils produced an immune response to the cancer cells, so we could make the tumour cells visible to the immune system, critical for any vaccine. Whenever I say good news, there's always some bad news. And the bad news is that we released all of these devils, nearly 100 of them, and the good news is that they found their way out into the wild. They survived really well, but unfortunately, a lot of the devils that we retrapped a year later, that had been immunised, came down with the tumour. So even though we could induce an immune response, it wasn't fully protective. So the tumours did develop. So we have to sort of go back to basics and try to work on improving that particular vaccine. So we found that the devils immunised, produced an immune response, were not protected from the infection, but there was some evidence of an immune response. If we have a look here, this slide here shows the tumour cells. This one here shows the immunised devils. This one here shows the incumbent devils. That was the wild devils that were already there that had tumours. And basically what that shows is that the immunised devils had evidence of an immune response, whereas the non-immunised devils didn't. So something is happening, but it's not good enough to protect. So the goal now is to promote that response even better to make a fully responsive vaccine, which we're working on. Now, to understand what might be happening is that we looked at the tumours themselves to see what was happening within the tumours. So we had tumours from the non-immunised devils and tumours from the immunised devils and analysed them. Now, remarkably, we could treat these devils with tumours with immunotherapy, putting in MHC positive cells, and that produced a response. That the devils that were immunised, they recovered. So we know that we can produce an antibody response, an immune response against the tumour cells, and we know that immunotherapy can cause the tumours to relapse, but in the wild we just can't get it to work as it should. So we analysed these tumour cells, and again, ran it through this sequencer, analysed thousands and thousands of genes, and produced what's called a heat map, and analysed all the genes that are expressed just to see what was expressed within the tumour. And there's thousands of genes. So the research staff sit on their computers hour after hour analysing it. And it was quite convenient because during COVID, no one was allowed to work in the laboratory, so everyone had to sit at their computers. So this sort of, the students didn't have an excuse not to work. They could use their computers. And again, these are the genes that were downregulated. These are the genes that were upregulated. You don't really need to know what they are, but to summarise, and this is what I find incredible, is that the DFD1 tumours in the immunised devils, where there was some evidence of immune response, they underwent a process of de-differentiation. They changed. And they changed from their myelating phenotype, remember DFD1 is a myelating DFT cell, to a mesenchymal phenotype. Remember, mesenchymal was a type of tumour that DFT2 was. So And this is an example of what we call plasticity. You can swap from one type to another. So this was quite remarkable. And just to show you that diagrammatically, that's a devil. And in here we have a tumour, and here's some immune cells. Because, remember, DFT1 is invisible to the immune system, nothing's happening, and the tumour develops into a myelating DFT1 tumour. The immune response won't respond because they don't express MHC, so they're invisible. This is an example where we immunise a devil, an immune response is happening, but what's happening is that the immune response is producing cytokines which, for some reason, alter the phenotype, the type of Schwann cell, and they become a mesenchymal Schwann cell. And the mesenchymal cells aren't easily responded to by the immune system. And there's a process which we call epithelial to mesenchymal transition, EMT, which is how cancers, particularly lung cancer, can avoid immune responses. And it's quite common in cancers. They go under this plasticity, they swap from one to another. So not only do we have to target these cells here, we have to target the mesenchymal cell. So there's obviously a little bit of work to do. So it's at Schwann cell plasticity. DFT1 cells go from the myelating in response to immune response, and they go to the epithelial or mesenchymal transition, DFT1. And remember, these repair Schwann cells are DFT2, so that's just a difference in that differentiation. So that was fairly technical. Now, a devil dilemma, is there hope? I've just said that we can produce a response and not protective. They don't produce MHC, they swap from epithelial to mesenchymal. Do we give up? No, we don't give up. There is some exciting evidence in that we have discovered in the last 5 years or so more than 20 devils that have had tumours. We have teams that go out, they trap devils, and then they release them, trap them again 3 months later, release them. Some devils that had early signs of tumours, trap them 3 months, the tumour's gone. So there's evidence of wild devils responding to the tumour cells, responding to the cancer. So there's some evidence of a natural immune response. Only in a very small number of devils, when you think of 53,000 down to 18,000, and only 20 or so have recovered. It's not a large number. But also work in our colleagues at University of Tasmania have shown that the genes are slowly changing. So there's a sort of, they call it a rapid evolutionary response, or maybe it's a selection. So there is something happening in the wild where the devils seem to be adapting to the disease, and whether or not they'll adapt quickly enough to protect themselves, we don't know. So the devil dilemma is basically, do we interfere with science, or do we let nature take its course? If we let nature take its course, they could become extinct. If we let the science, we could be altering the course. It's one of those dilemmas that will never be right. I think science, other people think nature. I think science because I'm totally biased. But if we see enough devils respond in the wild, I'm quite happy for them to recover. Okay, so why are devils susceptible to transmissible cancers? Why devils? Why not other animals? Why Tasmanian devils? People ask us all the time. So we thought, well, we better try to find out. And we put together this theory, which is a theory, but we have evidence to support it. And again, a scientific cartoon. This is a tumour cell, a Schwann cell tumour cell. And what's important here is these two molecules, MHC, remember MHC is the molecule that the immune system recognises, and there's this other molecule called ERB-B3. You don't need to remember that. That is a oncogene which drives the proliferation of the tumour cells. Cancer cells divide because oncogenes are turned on. Colleagues of ours in Austria have discovered this molecule ERB-B3, which allows the cells to proliferate. It also turns off MHC. So very early on that very first cancer, we had these tumour cells develop. The Schwann cell became a cancer, and it developed into a tumour. It expressed MHC as it would, but was fairly low, and it could be transmitted to local devils. Tasmanian devils have a low genetic diversity. In other words, they're pretty much inbred. Their genes are fairly similar. And in local areas, that genetic diversity is lower because they're mating amongst each other. So what we believe happened is early on, this cancer, which is represented by the blue colour, would only be transmitted to devils that were genetically similar. Right, so they went into that devil and that devil. And then something happened in these cancer cells here. The ERB3 increased its expression, they became more proliferative, MHC was turned off, and then they could be transmitted from genetically similar to, this one here is mutated, they can go from genetically similar to genetically dissimilar. So early on, the transmission was between devils of similar genes. Modification happened to the tumour cells, and that could be transmitted amongst the whole population. So they are transmitted throughout the devil population, basically because they turn off their MHC and they may produce immunosuppressive cytokines. Okay, so that's why we believe devils are susceptible to transmissible cancers. The next question is, why are they susceptible to Schwann cells? Why not other cells? I've got no idea, but this is my theory, is that they bite each other on the face, all right? When you see a Tasmanian devil, it's got lots of whiskers, right? Whiskers connect to peripheral nerves. Peripheral nerves are coated by Schwann cells. So on the face of a devil, there's lots of peripheral nerves, a lot of Schwann cells. When they bite each other, they cause damage. All right, when you cause damage, you need to have some sort of repair. And the repair Schwann cells promotes the growth of the tumour cells. The face is a Schwann cell which area? That is an area where Schwann cells like to grow. If you put Schwann cells in the rump, it wouldn't grow so well. But in the face, it's an environment very conducive to growth of Schwann cells. The MHC is very low on the Schwann cells by their very nature. They get transmitted for devils of similar MHC type, then they mutate and can be transmitted amongst non-similar devils. It basically is just a perfect storm of a whole lot of things happening together. It's an accident of nature. Somebody asked me, would this happen again? And I said, no, it's such a freak of nature. So many things had to happen together that would never happen again. And I was wrong. So I'm not going to make any more predictions. We're now preparing for DFT3, DFT4, and you'll probably be familiar with this virus is going well, how that's mutating, and that's similar to what may be happening here. Who knows? Okay, let's change directions, and I'm going to introduce you to another topic called extracellular vesicles. Something you may not have heard of, but something that's going to change medicine in the future. Extracellular vesicles are vesicles, little membrane structures that are released from cells. Now, a bit of its membrane will bud off, and they can be found circulating in the blood. And often a diagnosis of cancer can be done by identifying these proteins, these membranes in the blood, rather than actually having to take a biopsy. So they're found in the blood. They're also important for cell function. One of their functions they're important for is allowing cells to move from tissue to tissue, a process known as metastases. Now, a tumour will start in one area, will invade the tissue, get into the circulatory system, get into the lymphatics, and metastasise to another tissue. These extracellular vesicles allow that to happen. So we thought, could we use these molecules as a differential marker to differentiate between DFT1 and DFT2. In the wild, with a tumour like this, you can take a biopsy, take it back to the lab and analyse it. But if the tumour hasn't ulcerated, these have ulcerated, if the skin hasn't broken, we're not allowed to do biopsies because you're affecting the process. So you don't know whether it's DFT1 or DFT2, and that has important implications because you don't know what sort of tumour they've got. So if we could do a blood test to identify it, that would be a major advantage. And as I said, extracellular vesicles can be found in the blood. So we started looking for these extracellular vesicles, looking for these little structures in the blood. And basically, we've got cell lines growing of these different cancers. So this is a DFT1, a DFT2, and Fib, it's not a lie, it's a fibroblast, representing a normal cell. Grew those cells up and basically extracted the extracellular vesicles and ran them through a mass spectrometer just to analyse what proteins were present. Again, a very important machine that can identify different proteins based on their size, structure, isoelectric point, charge, all these sorts of things. And then to see what we could find. And this is an example of those extracellular vesicles. It's a very messy preparation. This is an electron microscope, but they are about 150 nanometres in size. Now 1 nanometre is 10 to the 9 metres. 1,000 nanometres will equal a millimetre, so they're extremely small. That's why you need an electron microscope. Okay, so this slide, I'll now run you through it to identify these extracellular vesicles. This graph on the x-axis, the horizontal axis, is comparing DFT2 with DFT1, a ratio of the amount of these proteins, these extracellular vesicles we found in the blood. So it's a ratio. If we go to the right, there's more DFT2 than DFT1. If we go to the left, there's more DFT1 than DFT2. And on the y-axis, the vertical axis, it's a probability that this is a real result. And what we look for were those proteins there which were more highly expressed in the DFT2 devils with DFT2 tumours. We found they're more common in the blood of devils with DFT2 than in the blood of devils with DFT1. So we analysed those, did another heat map, and basically what that tells us is that we identified a whole range of molecules which were important for the metastases process or important for the migration or adherence to other tissues, and one molecule stood out, and that molecule is called tenascin C. I hadn't heard of it either, but it's a molecule that is involved in the cells migrating and adhering to tissues. But we then looked at it and said, well, maybe this molecule can differentiate between DFT2 and DFT1. So we compared devils with healthy devils, diseased devils with DFT1 and DFT2. So that graph there on the x-axis, that's a healthy devil, that's a devil with DFT1, and that's a devil with DFT2. And up the y-axis is the amount of protein present. And what you can see is that the healthy devil and devil with DFT1 had the same amount of this molecule. But DFT2, this one here, much higher. So now we've got a marker, a blood marker, which can be used to distinguish DFT2 from DFT1. It's going to make enormous implications in the diagnosis. And that marker is a mesenchymal marker. Mesenchymal, you remember, is what DFT2 was, a mesenchymal Schwann cell. So it's sort of, thankfully, it's all fitting together quite nicely, that DFT2 is of the mesenchymal phenotype. So then we got really ambitious. We thought, can we use these extracellular vesicles as a preclinical marker? In humans, a preclinical marker might be the PSA test for prostate cancer. That's not very good. Maybe the pap smear, maybe the poo poke post test, the occult blood test. There's not very many, so we thought we'd be really ambitious and see if we can find something in devils that may predict whether a devil's going to be DFTD. So we got the serum from a number of devils, healthy devils and diseased devils, and devils that were presumed to be latent devils. What is a latent devil? A latent devil was a devil that was trapped, it was healthy, and the next trapping, 3 months later, it had a tumour. So during that period between those trappings, the tumour developed. So at that first time, we're predicting that it had the tumour, but it wasn't visibly detectable at the latent period. And in COVID, we have that incubation period. Now, I have to quarantine for 2 weeks or whatever. So that's an incubation period. Very similar to what we've got here is the latent period. So I compared the serum from all of these, very preliminary, but we found one molecule that stood out. this molecule here called Cath3. And this, we haven't published this yet, so this is very new, and you may never hear of it again, but is very promising. This molecule Cath3 is very low in healthy devils. The green represents healthy devils, and is increased in latent devils, those apparently healthy-looking devils that later on developed DFT, and this is DFTD, DFT1, and similar to devils with DFTD. So is there a preclinical marker to identify DFT1? Maybe. But who knows? But I find this really exciting. I still can't believe it, but I think it's really exciting, because then you can screen devils without having to take blood, without having to incubate them and put them in insurance populations for a period of time. Okay, so back to the vaccine. What's the next strategy? The final double devil dilemma is to identify the tumour antigen. We used whole tumour cells, but now we're working on trying to find the particular antigen that the immune system recognises. Now, those of you who are familiar with the COVID vaccine, you're immunised not against the whole virus, but against the spike protein, a particular part of the protein. So we're trying to identify a particular part of the cancer cells that will induce an immune response. So that's what the group's working on at the moment. But we also have to work out a way to deliver the vaccine. That first trial, we had devils in captivity. We could catch them, immunise them and release them. Obviously, we can't do that in the wild. We can't call devils in, immunise them and tell them to come back 2 weeks later with their certificate of identification. So we have to work out another way of doing it. And one potential way is with an oral bait. Now, oral baits have been very successful in reducing the frequency of rabies in Europe. They have dropped these baits from aeroplanes and helicopters. So we thought we could, this is the Andy Flea's work, try to develop a way in which the devil can take baits. If you put baits on the ground, what's going to take it? Those damn possums. They'll take the lot. So we had to work out a way of which you could get for the devils to take the bait, but not the possums. And they developed this little trap, which is the trap's there, where the devil has to climb up and put its snout into the trap and pull the bait out. Possums can't get to it, mice can't get to it, cats can't get to it, and so we've got a way in which the devils can get it. We put little cameras on there, and we can watch the devil take the trap, so they could be distributed. But then you had to work out what the devils like to eat that possums don't like. And so we've tried fish oil, we've tried all sorts of things, and we're working out which the devil favours, and next year we should have some more results from that. But they do like fish oil. They do like smelly stuff, so they'll take the baits quite well. Okay, to summarise that, and I'll highlight these points. Devil Facial Tumour Disease comprises 2, not 1, but 2 independent, separately derived transmissible cancers. I didn't tell you this one. The first one was derived in a female, and to balance the gender, the second tumour derived in a male. They are both cancers of Schwann cells. The pathogenic agent is the cancer cells. They are neurocane endocrine cancers of Schwann cells. And DFT1 is of a different phenotype than DFT2. DFT1 is of the myelin-producing type, but remember it can alter into the repair phenotype in response to the immune system. DFT2 is a repair-type Schwann cell. And the reason why the tumours can be transmissible is that the immune escape is they don't express MHC. Early on, the tumours transmit between devils of similar genetic type, then they mutate, lose MHC, become invisible, and they can be transmitted through any devil of any type. Why Schwann cells? That perfect storm, biting, Schwann cell rich, transmitting the tumour cells from face to face. Schwann cells like to grow, repair Schwann cells, all those sorts of things. All things happening together in a perfect storm. We've got a blood test which identified tenascin C, which can identify DFT2 from DFT1. DFT2 expresses it, DFT1 doesn't, and that's a mesenchymal marker which is consistent with the mesenchymal type of DFT2. And the good news is that we have a vaccine that produces the immune response. Some wild devils are showing evidence for resistance, so we know we can produce an immune response. We know it can happen in the wild. We can induce it artificially, but our vaccine needs a lot of tweaking to make it much, much more effective. And I'll finish off with a quick summary these are the people working in the lab. All of them have been supported by research grants, Australian Research Council, but importantly, the Save the Devil program appeal is always looking for donations, which comes to the research. I'd like to acknowledge Devil ambassadors John English, who you recall was promoting the Devils for a number of years. Now it's Bonnie Sveen, out there trying to get public interest in protecting the species. And if you want to know what you can do, one thing you can do is encourage everyone to slow down at night. Roadkill is a real problem. I get embarrassed when I have visitors and we see how much roadkill we have on our roads, and it is a big risk for devils. Black devils at the night time, black roads, they're very hard to see. So I'll leave it there and thank you very much for your attention. I can answer questions.
If anyone has any questions, please signify. Please feel free to ask. The professor's very happy to answer.
As long as they're easy, yeah.
A beautiful 3 months between catching a devil and then an interval of 3 months between catching an apparently normal devil and finding it develops the tumour. Do you know or have any idea of how long between the bite and the development of the development of the tumour, what that interval is? Presumably it's less than 3 months.
We don't know. We don't know because if you get 5 tumour cells or 5 million, it's going to be different. Depends on the bite. It's going to depend on the actual tumour cells themselves, how viable they are. So it ranges from 6 to 12 months. It could be as low as 3, but it could. With some we think we went more than 12 months. So it's sort of about six months of the period from when you get the bite and a visible tumour. Once it's visible, it just appears to grow very quickly. But it's a fairly long incubation period.
What about the bite and the abnormal blood test?
Yeah, well, we don't know when the bite happened. So what will happen is that there'll be a healthy devil that will take a sample, but we don't know when that devil was bitten. Might have been a month before, it might have been after. And if we could keep an eye on the devil, see when it bit and then take blood samples regularly, we might know, but we just don't know when it happened. The field work is very labour intensive, and Tasmanian winters are really bitter. So it's hard to get volunteers to go out in the winter time, and it's very expensive.
You spoke at one stage of 2 paths seeming available for going forward. One was a scientific intervention path, which you favoured, owing perhaps to bias. I'm a philosopher, not a scientist. I have no such bias. I also favour that. The other was just a natural immunity build-up. I would have thought, and I wanted to comment on the natural immunity build-up path in a couple of ways. One was to think it's not a very good path, because I would have thought that it would lead to a not very genetically diverse population becoming even less genetically diverse in non-cancer related ways, which might be a problem in itself. So I wanted some comment on that. The second, looking at it though, here you have individual devils with no intervention from guys like you managing to resist things. Has anyone, or I assume you would be, but you didn't comment down this path, Has anyone had a look at these guys to try and work out what it is they're doing that you might be able to simulate with an intervention?
Yeah, both very good questions, and thank you for those questions. The question about letting nature take its course and the genetic diversity is that you may be selecting for those particular devils that can respond in a certain way, but if another disease comes across, they don't have the diversity to protect them against that. So we're concerned about that. Devils have a low genetic diversity from the start. The other debate that we're having is that there's devils in the insurance population. The idea was that when the original idea was devils in the insurance population, when the devil population almost became zero, they'll be reintroduced back into Tasmania. Well, that's not happening because the population's gone down and it's sort of plateauing and goes up and goes down again because of the contact nature of it. Now, those devils in the insurance population were collected 10 years ago. So if you like, they have the genotype of those devils 10 years ago, whereas the devils at the moment, if the evolution is happening, they've got the genotype of the more resistant devils, if you like. So if you bring those devils back, you might be reintroducing a sensitive genotype. That's one of the possibilities. The question about what are these devils that have responded, what's special about them, they have been analysed and the work's coming out, and that's part of that rapid evolutionary process, analysing all of the genes to see what's expressed before and after to try to look at. But with just a few animals, and it's very hard to get a good indication. But yeah, that's one area that has been looked at, comparing the devils that seem to have recovered, devils that haven't. Try to look at the genes before the tumour was here, genes were after, to see what has been happening. And there's lots of interesting results, but it's really, really hard to dissect the specifics. But yeah, thank you. They were both good questions.
Hi. Over here.
Sorry.
Just wondering, with regard to roadkill, so being carnivores, so they bite during mating, bite during feeding. Do they eat their own?
Yep.
So transmissible in that regard as well. So roadkill.
Yep.
Yeah.
Devils will eat each other. Yep, they certainly will. And they really like the nice soft cancer material that will go first. They bite each other, they eat each other, they clean things up completely. And well, the reason why they get the tumours occur in the mouth is that devils eat absolutely everything. They'll crunch through bones. They'll eat. We've found quills. Now, next time you go walking, have a look for devil scats and see if you can find any quills. They'll eat echidnas, and the bones will cause scratches in their oral cavity. If they bite a tumour, the tumour will get into the oral cavity and start to grow. It's just their behaviour. If we could stop all this behaviour, it would stop the tumour, but then they wouldn't eat, they wouldn't reproduce, so we wouldn't be any better off. But roadkill is something that we really worry about, because apart from the tumour, the roadkill is their next biggest threat.
I was just wondering, has anyone done any research from where the first area was where the facial tumour disease outbreak was, whether there was anything environmental there that actually caused this first outbreak to happen? Has anyone gone back there so much later to have a look and see if, with all the research you've done now, to tie anything in with that?
Yeah, that's a good question, and often comes up, and there's no easy answer, because if you go back there 20 years later, things have changed. If we didn't have this second cancer, we might think that there might be something special about that particular area. The genome has been sequenced, and a lot of carcinogens have a signature mutation. And there hasn't been any signature mutations identified in the tumours. So there's no real evidence of a carcinogen causing that first cancer. We don't know. I'm not saying it couldn't have happened. It's possible. But what we have to recall, it only happened in one animal and then it transmitted. DFTD2 started off in one animal and got transmitted. The first one was up Mount William, near Waterstone Point. The second one happened down near Cygnet in the Channel Peninsula, both totally different areas, so you wouldn't think there would be something particular common. I have a theory that is probably not right. It's probably definitely not right, but up in that particular area, the forestry was very important, and I'm not saying that they caused the tumour. The area was cleared, the trees were protected, poison was put out there, lots of possums died. So that made a fantastic food source for devils, so there wasn't the competition. Now, I hate to tell you, all of us being of that age, the longer you live, the more likely you are to get cancer. Right, it's just a build-up in mutations. I suspect that devils live in the wild. For a species that vigorous, that aggressive, that handsome, that strong, only lives 4 to 5 years. Very short time. I suspect that because there wasn't this competition, that a devil lived maybe 6, 7, 8 years, and during that time, it was just a build-up of age, mutations, and that cancer developed. Devils are prone to cancers. Most of the devils in the zoos that live for a long period of time, they die from cancer. So they are prone to cancer. It'd be nice if it was an easier answer to that one, then we'd be able to respond to a lot of critics. Oh yes, we know it exactly, but because we don't know, you can argue both ways, and you don't get a resolution, and no one ends up happy.
Well, thank you very much indeed, Professor Greg Woods, for giving us such a fascinating insight into this unique problem of Devil Facial Tumour Disease. I found it very enlightening indeed, and I think you've explained it at a whole range of levels as to why it occurs, why it's so unique, and we're very grateful. Thank you. We'd like you to have a souvenir certificate of your attendance here with a brief history of the society on the back. And
I am a member, by the way.
Well, there you are. You're in Coles to Newcastle. And one of our publications that I hope you find as interesting is your Devil Facial Tumour Disease. Thank you very much for being our guest today.